Welcome to the deep dive. Looks like we're diving into some immunology today, specifically antibodies.
Yeah, that's right.
I know it can get pretty complex, but it's a fascinating area for sure.
Absolutely.
We're going to try to break it all down for you today.
Yeah, help you navigate this complex world.
You've been spending some time with chapter 5 of that immunology textbook, right?
Yeah, that's the one.
The one that really delves into the intricacies of these antibodies and antigens?
That's right. It's quite a deep dive into that world.
Yeah, and hopefully by the end of this deep dive with us, you'll have a better grasp on how these things are structured, how they work, and how they're so diverse.
Yeah, their diversity is really remarkable.
It's almost like, a microscopic army with specialized units for every possible enemy.
That's a great analogy.
Right. It's pretty wild.
It is, and the story of how scientists actually discovered these amazing proteins is pretty fascinating.
Oh, tell me more about that.
It's like a scientific detective story.
Okay.
So early research says they were studying this terrifying disease called diphtheria, which was especially dangerous for children.
Oh, wow.
And they discovered that there were certain proteins in the blood that could actually neutralize the diphtheria toxin.
So that's why they were initially called antitoxins, right?
Exactly, because these seemed to be working against these toxins.
Interesting.
But then, as scientists dug deeper, they realized that these proteins had a much broader reach. They can actually target a whole range of substances, not just toxins.
Wow.
So that's where the term antibody came from.
Okay.
And the substances they targeted became known as antigens.
So they're like the ultimate multitaskers of the immune system, really.
That's a great way to put it. Yeah, very adaptable.
Yeah.
And to understand how they actually work, we need to look at their structure.
Okay.
And you can think of an antibody as sort of a Y shape,
All right.
with two identical light chains and two identical heavy chains,
Okay.
forming like the arms and stem of the Y.
Okay, so a Y shape, I'm picturing it.
Yeah, exactly.
What about these light and heavy chains? What's special about them?
Well, these chains are made up of repeating units that we call immunoglobulin domains.
Immunoglobulin domains.
Yeah, or Ig domains for short.
Okay.
And these domains are like modular building blocks.
Okay.
They give antibodies their unique properties.
So these ion domains are what make antibodies antibodies.
Exactly. But what really empowers them is the distinction between the variable and constant regions within these chains.
Okay.
You can think of the variable regions, or V regions, at the tips of the Y as like highly specialized sensors.
Okay.
Each one is uniquely shaped to recognize a specific antigen.
Ah, so each antibody has its own unique fingerprint in a way.
Yeah, that's a great way to put it, a unique fingerprint that allows it to latch on to a specific target.
Got it.
And then you have the constant regions, or C regions, which form the stem of the Y.
Okay.
They determine how the antibody actually deals with the threat once it's bound.
So the V regions recognize and the C regions act.
Exactly. A perfect tag team.
I like it.
And scientists actually discovered that if you cleave an antibody with enzymes,
Cleave meaning cut.
yeah, like cutting the Y shape, you can separate it into two fragments, the Fab fragment and the FC fragment.
Interesting.
The Fab fragment, containing the antigen binding sites,
Okay.
is responsible for grabbing onto the antigen, and the FC fragment carries out those effector functions we talked about.
Okay, so it's like a division of labor.
Yes, exactly.
Fab for binding, FC for action.
You got it.
But this Ig domain structure, is that unique to antibodies?
That's a great question.
Yeah.
And what's fascinating is that the Ig domain structure is not exclusive to antibodies.
Oh, really?
You find it in many other immune system proteins, which suggests a shared evolutionary origin, a common ancestry.
So it's like a blueprint that's been adapted for different functions.
Precisely.
That's pretty cool.
Yeah,
But let's go back to those remarkable variable regions for a minute.
sure.
How do they generate such a mind-boggling array of different antigen-binding sites?
That's the key question, right?
It seems like there must be a huge number of possible combinations.
Absolutely. And that diversity comes from three small stretches within the V regions called hypervariable regions,
Okay.
or CDRs for short.
CDR's got it.
And these CDRs, particularly CDR3, exhibit the greatest variability.
Okay.
They form the heart of the antigen binding site.
So it's these tiny regions that determine the incredible specificity.
It is amazing, isn't it?
Yeah, it is.
Think of it like this. The CDRs are like fingers protruding from the antibody,
Okay.
and they interlocked with the antigen like a 3D puzzle.
Oh, okay.
So the antigen fits snugly into the space created by those CDRs.
Interesting.
And the tighter the fit, the stronger the interaction.
That makes sense.
Now, you mentioned earlier that antibodies can be either polyclonal or monoclonal.
Yeah, what's the difference between those?
Well, think of it like this. When you get infected or vaccinated, your body produces a diverse mix of antibodies
Right.
that target different parts of the antigen.
Okay.
That's what's called polyclonal antibodies.
So polyclonal antibodies are like a mixed force targeting the antigen from all sides.
Exactly, like an army with various specialized units attacking different parts of the enemy.
Okay, so what about monoclonal antibodies then?
Monoclonal antibodies are more like having a highly specialized unit with laser focus.
Okay,
They're all identical,
right?
produced by a single B-cell clone,
Uh-huh.
and they target one specific epitope on the antigen.
Ah, so monoclonal antibodies is like snipers going after one specific target.
Exactly, a very precise weapon.
Interesting.
And the ability to actually create these monoclonal antibodies was a real game changer in medicine and research.
I bet. That's incredible.
Yeah, it's pretty amazing.
But how do scientists actually create these monoclonal antibodies?
It's a pretty brilliant technique, actually.
Okay.
Imagine fusing a B cell, which is the cell that produces antibodies, with an immortal myeloma cell line.
Okay.
Now, myeloma is a type of cancer.
Right.
So this hybrid cell, which we call a hybridoma, can produce antibodies like a B cell,
Okay.
but it also keeps dividing like a cancer cell.
So it's like creating a tiny antibody factory.
That's a great way to put it. A tiny factory that churns out a continuous supply of identical antibodies.
Wow.
And because each hybridoma originates from a single B cell, it only produces one specific antibody.
Okay.
That's how we get monoclonal antibodies.
That's really remarkable.
It is.
It's like harnessing the power of the immune system in a very targeted way.
Exactly, to produce customized weapons against disease.
Wow, that's amazing.
It really is.
Okay, I think we've covered a lot of ground here already, going from the basic structure of antibodies
Yes,
to this revolutionary power of monoclonal antibodies.
a lot to take in.
It is. This is all fascinating stuff. I think we need to take a little break here and let all this information sink in.
That sounds good.
When we come back, I want to delve a little deeper into the different classes of antibodies
Okay.
and how they each play these unique roles in the immune response.
Sounds good.
Stay tuned. Welcome back. It's amazing how scientists figured out how to create those monoclonal antibodies, right?
It really is, yeah.
It's like they're hacking the immune system in a way to develop these very targeted therapies.
Yeah, absolutely. Harnessing its power.
But let's shift gears a bit and talk about the diversity of antibodies themselves.
Okay,
Because they're not a one-size-fits-all kind of defense, right?
that's right.
They come in different classes, each with its own, unique set of skills.
Yeah, like different tools in a toolbox.
Exactly. So what are these different classes called?
We call them antibody isotypes.
Isotypes, okay.
And there are 5 main ones to keep in mind.
Okay.
IgA, IgD, IgG, and IgM.
Wow, that's a lot of IDs.
It is, but don't worry, we'll break them down.
Okay, good.
Each isotype has a distinct heavy chain constant region. Remember those action regions?
Right, the ones that determine how the antibody actually does its job.
Exactly. So different isotypes, different functions.
Okay, tell me more about these different isotypes.
All right, well, let's start with IgG.
Okay.
That's the most abundant antibody in our blood.
Oh, wow.
It circulates for weeks, thanks to a special receptor that protects it from degradation.
Interesting.
You could think of it as the workhorse of the antibody world.
IgG, the workhorse.
Yeah, great at neutralizing microbes and toxins.
So it's like the long-lasting protector, constantly on patrol.
Exactly. Always there to keep us safe.
What about IgA? What does that one do?
IgA is known as the guardian of mucosal surfaces.
Mucosal surfaces, so like what?
Think about all the areas of your body that are exposed to the outside world.
Okay, like,
Your gut, your airways.
Right, right.
IgA is found in high concentrations there, acting as a first line of defense.
Oh, so it's like the border patrol preventing invaders from getting in.
That's a great way to put it.
And it's also passed from mothers to their babies through breast milk, right?
You got it. Providing that crucial protection in those early months.
That's amazing.
It is, and it's just one of the many ways a mother's immune system helps protect a newborn.
Wow, incredible.
Now let's talk about IgM.
Okay, IgM.
This is often the first antibody produced during an infection.
Ah, so it's like the rapid response team.
Exactly, the first one on the scene.
So IgM is there to quickly control the threat.
Precisely.
Why is it so effective at that early stage?
Well, IgM has a unique structure.
Okay.
It's a pentamer.
A pentamer, what's that?
It means it's made up of 5 antibody units joined together.
5, wow.
Yeah, so that gives it 10 binding sites.
10, that's a lot.
It is. It allows it to grab onto antigens with incredible strength.
Wow, so IgM is like really good at grabbing onto those invaders.
Yeah, we call that high avidity, that ability to bind with such strength.
High avidity, got it.
So you see, IgM is a powerful force in those early stages of infection.
Okay, so we've got IgG, IgA, and IgM.
Yep, those are the heavy hitters.
What about IgD and IgE? What do those do?
Well, IgD is a bit of a mystery, actually.
Oh!
It's found on the surface of B cells, but its exact function is still being investigated.
So it's the one with secrets.
You could say that, yeah.
Interesting.
Scientists believe it may play a role in B cell activation
Okay.
and maybe regulation, but more research is needed.
All right, the mysterious IDD.
Now, Ig, that one's famous for its role in allergies.
Oh, right, IG, the allergy antibody.
Exactly.
I know some people who aren't too fond of IG.
I can understand why it binds to mast cells.
Mast cells.
Those are the cells that release histamine and other inflammatory mediators.
Right, those are the things that cause all those allergic reactions.
Exactly. The sneezing, the itching, the watery eyes.
Not fun.
Not fun at all. But, it's important to remember that Ig also plays a role in protecting us from parasites.
Oh, right, so it's not all bad.
Nope. Just like everything else in the immune system, it's all about balance.
Okay, so we've got all these different antibody isotypes, each with its own specialized function.
That's right.
But how does our body know which one to produce in response to a specific threat?
That's a great question. And it brings us to one of the most fascinating aspects of antibody function.
Okay, what's that?
Heavy chain isotype switching.
Isotype switching, what is that?
So when a B cell is first activated, it usually starts by producing IgN.
Right, the first responder.
Exactly. But as the immune response progresses, that B cell can actually switch to producing a different isotype.
Wait, so a B cell can change the type of antibody it's making?
It can. It's pretty amazing.
That's incredible. How does that even happen?
It involves some clever gene rearrangement.
Gene rearrangement.
Yeah, essentially the B cell can change the constant region of the heavy chain
Okay.
while keeping the variable region, the antigen specificity, the same.
So it's like changing the tool but keeping the target the same?
Exactly. Adapting to the challenge at hand.
But how does the B cell know which isotype is the best tool for the job?
It receives signals from other cells in the immune system,
Okay.
like T helper cells and dendritic cells.
Uh-huh.
These cells release signaling molecules called cytokines,
Cytokines, right?
which basically act like instructions, telling the B cell which is a type to produce.
So it's like the immune system has its own internal communication network?
Precisely. A very sophisticated one.
Different cells coordinating the attack.
Exactly. It's remarkable.
Wow, isotype switching? That's amazing.
It is a key example of how adaptable and sophisticated our immune system is.
So isotype switching is one way that antibodies can change, but there's also that other thing, affinity maturation, right?
That's right. You're on a roll.
You mentioned that antibodies can actually improve their binding strength over time.
They can, and it's a remarkable process.
It's almost like they're evolving, getting better at their job.
It is a bit like evolution, actually.
So how does it work? What's happening?
Well, as the immune response progresses, those B cells undergo something called somatic hypermutation.
Somatic hypermutation.
Yeah, try saying that 3 times fast.
I don't even want to try.
It's essentially a process of rapid mutations in the genes that encode those antibody variable regions.
Right, the regions that determine antigen specificity.
Exactly. So those fingerprints we talked about are getting constantly tweaked.
So the B cells are experimenting, trying to find a better fit.
You got it. And the ones that bind best, they're the ones that survive and proliferate.
Survival of the fittest, basically.
Exactly. The ones with lower affinity are outcompeted.
It's amazing how the immune system constantly refines its attack,
It's truly remarkable.
becoming more and more effective over time.
It is, and this process of affinity maturation combined with isotype switching, it just highlights the incredible adaptability and precision of our immune system.
All right, so we've covered a lot about antibodies.
They're fascinating.
But it's important to remember that they're only half of the equation.
That's right.
To really understand their role, we need to understand the targets they're going after.
The antigens.
Exactly. We've been so focused on the antibodies that we've almost forgotten about the other side of this immunological dance.
It is a dance, a complex interplay.
So let's talk about antigens.
Okay, let's do it.
What exactly are they, and what makes them such worthy adversaries for our antibody army?
That's a great question, and that's where we'll pick up next time.
Okay, we're back and ready to give those antigens their due.
Right, it's time to shine the spotlight on the other half of this immune system battle.
We've spent a lot of time exploring the incredible world of antibodies.
Yeah, those tiny warriors.
But they're only half the story.
Absolutely, we need to understand the targets they're designed to combat.
Exactly. So let's talk about antigens.
All right, let's dive in.
It seems like they can be almost anything: viruses, bacteria, toxins.
Yeah, the list is pretty extensive.
What exactly defines an antigen then?
An antigen is any substance that can be recognized by an antibody or a T-cell receptor.
Okay, so it's all about being recognized by the immune system.
Right, that's the key.
But that seems pretty broad. What kind of things fall under that definition?
Well, it's a surprisingly diverse group, actually.
Okay.
We're talking proteins, carbohydrates, lipids, nucleic acids,
Wow, that's a lot of different things.
even small molecules like hormones and metabolites. Essentially, anything that's foreign to our body has the potential to be an antigen.
So anything non-self could trigger a response.
Yeah, you could say that.
But I imagine not all antigens are created equal, right?
That's a good point. You're right.
Some must be more I don't know, potent at setting off alarm bells than others.
That's exactly right. The ones that really trigger a full-blown immune response, we call those immunogens.
Immunogens, okay?
Think of them as the real troublemakers, the ones that really get the immune system fired up.
All right, the real threats.
Yeah.
What makes some antigens better at triggering that response than others?
Well, size and complexity are definitely key factors.
So bigger's worse.
In a way, yes.
Okay.
Generally, larger molecules like proteins and polysaccharides are much better at stimulating a response than smaller molecules.
I see. So is it just a matter of being more visible to the immune system, like a bigger target's easier to hit?
It's a bit more nuanced than that.
Okay.
Remember when we talked about B-cell activation requiring the cross-linking of multiple antigen receptors?
Right.
Well, large molecules have multiple sites,
Uh-huh.
called epitopes or determinants, that antibodies can bind to.
Okay.
So a single large molecule can actually engage multiple B-cell receptors simultaneously.
So it's a stronger signal.
Exactly. It's like having multiple alarms going off instead of just one.
Right. That makes sense.
Now, smaller molecules,
Yeah, what about them?
they can be immunogenic too, but they usually need a little help.
Okay.
Sometimes these small molecules, called haptens, need to be attached to a larger carrier molecule,
Interesting.
like a protein.
So it's like a disguise,
Yeah, you could think of it that way.
making it look bigger.
Exactly, making the small molecule more noticeable to the immune system.
That's fascinating. And you mentioned earlier that antibodies don't actually bind to the entire antigen, right?
Right, good memory.
Just a specific region, the epitope.
That's an important point. Antibodies are incredibly specific.
Okay.
And that specificity comes from their ability to recognize those distinct epitopes on an antigen.
I see.
And these epitopes can vary quite a bit in their structure.
Okay, so different types of epitopes.
Yeah, you got linear epitopes, which are formed by a continuous sequence of amino acids.
Right.
Think of it like reading a sentence straight across.
Linear, got it.
And then you have conformational epitopes.
Okay.
Those depend on the three-dimensional structure of the antigen.
So it's more complex?
It is. It's like solving a jigsaw puzzle, where the pieces have to fit together in a specific way.
That's a good analogy. So antibodies can be specific for either type of epitope.
Exactly. They're versatile like that.
But how does this binding actually happen? What's going on at the molecular level when an antibody latches onto its target?
It's all about those non-covalent interactions.
Non-covalent interaction.
Yeah, those weak forces that hold molecules together without forming actual chemical bonds.
Right. Things like electrostatic forces and hydrogen bonds.
Exactly. Van der Waals forces, hydrophobic interactions.
I remember those from chemistry class, but it's hard to imagine how those weak forces could be strong enough to hold an antibody and antigen together.
Well, individually they might be weak, but collectively they can create a pretty strong interaction,
I see.
especially when multiple interactions are happening simultaneously.
So it's a team effort.
Exactly. Each weak force contributing to the overall strength of the bond.
The combined power of these weak forces creates a strong and specific interaction.
Precisely.
And you mentioned that the strength of that interaction is called affinity, right?
Yes, affinity is a measure of how tightly an antibody binds to its epitope.
Okay, and how do we measure that?
We often quantify it using the dissociation constant,
Okay.
or KD.
KD, got it.
The KD gives us an idea of how easily the antibody-antigen complex falls apart.
Okay.
So a smaller KD means a stronger affinity, a tighter bond.
The lower the KD, the stronger the interaction. Got it. But I also remember you talking about avidity.
Oh, yes, avidity.
How is that different from affinity?
Well, affinity refers to the strength of a single bond between an antibody binding site and its epitope.
Okay.
Avidity takes into account all the binding sites involved.
Right, because some antibodies have multiple binding sites.
Exactly, and some antigens have multiple epitopes.
So avidity considers the overall strength.
Yes, the combined force of all those individual bonds.
And I imagine that avidity can be much stronger than affinity, especially with an antibody like IgM with its 10 binding sites.
Absolutely, that's one of the reasons why IgM is so effective at those early stages of an infection.
It's like having 10 times the gripping power.
You got it. Even if each individual bond is relatively weak, the overall interaction can be incredibly strong because of avidity.
Wow, that's really interesting.
It highlights the importance of considering both affinity and avidity when we're talking about antibody-antigen interactions.
Okay, so we've covered a lot about how antibodies bind to antigens.
We have. It's a fascinating area.
I'm curious, what happens when you have a lot of antibodies binding to antigens? Does it just create a jumble of molecules floating around in the blood?
That's a great question.
Yeah.
When you have a lot of antibodies binding to antigens, they can form what we call immune complexes.
Immune complexes, okay.
These complexes can be small or large, depending on the relative concentrations of antibodies and antigens.
So it's all about balance.
Exactly. And if these immune complexes become too large, they can actually cause problems.
Oh, how
They
so?
can get deposited in tissues and trigger inflammation, leading to something called immune complex disease.
Immune complex disease? That doesn't sound good.
It's not. It's a reminder that even the immune response, which is essential for our survival,
Yeah,
needs to be carefully regulated.
that's fascinating and a bit humbling, isn't it?
It is. The immune system is so powerful, but it also needs to be kept in check.
Absolutely. It's a delicate
It
balance.
is a delicate dance.
And understanding that balance is key to appreciating the complexity and elegance of our immune system.
Well said. I couldn't agree more.
Well, I think we've done it. We've explored the incredible world of antibodies and antigens from their intricate structures to their complex interactions.
It's been quite a journey.
It has, and we've even touched on the potential consequences of those interactions.
Right. It's not always a straightforward story.
It's been a true deep dive.
It has, and I hope you've gained a newfound appreciation for these remarkable molecules and the intricate dance they perform to keep us healthy.
I know I have. And what's most amazing is that scientists are still uncovering the full potential of antibodies.
That's what makes immunology so exciting.
It seems like we're just scratching the surface of what these molecules can do.
Absolutely, there's always more to learn, and who knows what groundbreaking discoveries lie ahead.
Well said. And on that note, I think it's time to wrap up this deep dive.
Sounds good.
But remember, the journey of discovery never truly ends, so keep those curious minds engaged, and we'll catch you next time on The Deep Dive.